Okay. So hopefully everyone can hear me and see my slides okay. Welcome to the fourth iteration of this conversation series from the NMDC. Today we'll be talking to you about bioinformatic workflows, less the specifics of the workflows and more some of the challenges, some of the advantages, disadvantages of providing these types of containerized workflows and enacting them for microbiome research. So our agenda today is outlined here. I'll be giving you a brief introduction to the NMDC, the National Microbiome Data Collaborative, and some of the bioinformatic workflow efforts that we've conducted within our project, our pilot project. Today we have four expert speakers. I'll introduce them a little bit more later, but they're listed here. Of note, we will be hosting a large panel Q&A session. There's a shared note document that should be provided to you in the chat box. Feel free to enter all your information there, questions, or even your emails, etc. All of this will remain editable for 24 hours, and then this recording as well as the document will be viewable and available on the NMDC website. So I'll start by mentioning what most people already know. Those of you who've been in genomics or in microbiome research for some time, we've been undergoing a pretty dramatic shift in terms of the volumes of data that are coming out of newer technologies, in terms of all flavors of omics data. And they're quite varied in terms of the types of samples or the types of questions that people are looking into. And generally speaking, from a microbiome research perspective, you're interested in putting your data into perspective with everyone else's. But access to these data can be somewhat challenging. In fact, you can spend the majority of your time trying to find other people's data that you can use to compare with yours. And then there are further challenges in terms of how comparable are they truly. So our effort in the NMDC is to change this mantra to into a situation where you can readily search for other types of samples that fit your types of questions. If they're in a particular animal model, or if you're looking at a particular geographic environment, you can find other data sets that might be useful for comparisons, and then see what other data is available, and then access those data. So, apologies. The NMDC is a pretty large multinational lab effort out of the Department of Energy that is really trying to address this question quite broadly. So our main mission is to provide this fair access to data, this findable, accessible, interoperable, reusable data, and apply this to all flavors of microbiome data. We're focusing primarily on initial varieties of omics data generated by sequencing machines or mass spec instruments, but our goal would be even broader than that. We have a few publications, including one that pertains to bioinformatic workflows that has just been accepted, and you're also welcome to browse our main sites for any documentation or any of our work conducted so far. So we have a live portal website that's currently available. It's not populated with a tremendous amount of data. In part, that speaks to how difficult it is to link all the appropriate metadata for a sample or metadata for a data set to all of these raw omics data sets. But this is a vision of what we may want or may be able to do in the near future. The goal is to be able to search for any key term All these metadata are organized in a formal ontology. We have a schema that we've been refining and merging across a few different ontologies. If you're interested in ontologies themselves, there was a conversation as part of this series several weeks ago, and that recording is available on the NMDC website as well. Once you are able to find your data sets, you may want to narrow down which data sets you're actually interested in, and then you should be able to get access directly to those data sets. Right now, you can do all the searches without logging in or registering, but you'll need an ORC ID to register, and then you should be able to have access to downloading data sets and results that are provided in this website with all the requirements on how best to cite those data. So finding the data is certainly a key part of the NMDC pilot project. Another key component is to then be able to allow cross comparisons across different types of data or different environments so that you can put your project into perspective with others. So this requires a little bit more thought on the bioinformatic workflows, which is the topic of today's conversation. In the NMDC, we've focused on established workflows that have been working and running on a large number of projects at the Joint Genome Institute, as well as the Environmental Molecular Sciences Lab at PNNL, the EMSL. And so we've first tackled a variety of typical flavors of omics data. There are innumerable workflows available out there, so we've had to pick and choose to work with some of the ones that we're confident will give you some best practice answers. We've had to rewrite all of these, of course, because everyone's suite of tools or workflows or pipelines are all pretty much dedicated to their own environment. And our goal really was to make this a lot more interoperable. So allowing any user to grab these workflows and produce the same results that we would be able to produce either in the cloud or in any given institution. So in our hands, we've run a number of tests and evaluation or validation on these workflows. We've run them in a number of different venues and then compared results with at least 2 different data sets, which are also available through through our main GitHub sites. We also have made these workflows available in a couple of different venues. One is the NMDC Edge link that I've provided on this website. This is an interactive GUI run capability where you're able to upload your own data and process them yourselves. This will also be made available via the DOE KBase platform. So the workflows being done is certainly one thing, but we are also thinking about how to scale and how to manage these data writ large. So we've been developing an automation API that would allow us to register different sites for where the data is located, to register samples so that you can standardize and have all of your metadata be searchable. And then once you have those data, we have 2 initial nodes at NERSC, which is at the Lawrence Berkeley National Lab, that is coupled with the Joint Genome Institute, and so has access to those data. And so all of the metagenomic or metatranscriptomic workflows would execute there. And at the EMSL lab, we have a number of workflows that will work there for the mass spec data. These would then be registered, or all the data products, all the analysis outputs would be registered in the central store, which could then provide you access to those results as well and make some of those results searchable. So today's topic really revolves around workflow best practices, grand challenges, less to do with the nuances of each workflow, like which bioinformatics tools or what parameters or what thresholds do you use, but we have a broad range of expertise in our panel. We have Alicia Klum, who oversees a lot of the genome assembly and analysis at the Joint Genome Institute. I've known her for years. She's excellent and runs a number of these projects in the cloud as well as locally on their HPC. Kyle Chard, he's a research assistant prof at University of Chicago in the computer science department, and he co-leads the Globus Labs effort. There's a Globus platform that really enables large data sharing across anywhere in the world, and is being used by a large number of groups to enable data sharing and management. We have Steve Tsang at NIH. He's both part of NIAID, the National Institute for Allergy and Infectious Disease, as well as the Office of Data Science and Emerging Technologies, which I know is a mouthful. And he'll be talking to us about best practices at NIH for some of the national cancer efforts. And we have Danny Park at the Broad Institute. He's a group leader in the viral computational genomics effort, and he runs a large number of projects as well. He's been cornerstone in developing WDL and Cromwell, which is a workflow manager, which we use in the NMDC as well. So I welcome and thank our invited panelists, and I will let you take over and introduce yourselves a bit further, and as well as your ideas and any key messages you want to relay in terms of workflows. Oh, and I guess we'll start with Alicia.
Yeah, thanks. Okay, thanks. My name's Alicia, and as Patrick mentioned, I work at the Joint Genome Institute, and I'm going to be talking about leveraging containers and WDL for metagenomics workflows at JGI. Next slide, please. So if you aren't familiar, the JGI provides the global research community with access to advanced genome science capabilities for advancing solutions in bioenergy and environmental grand challenges. Next slide. We have a couple mission areas, including bioenergy, carbon cycling, and biogeochemistry. We do this through a couple of science programs and then capabilities, both instrumentation and computational. So today I'll mostly be talking about the metagenome program and how we do computational analysis on the sequencing data that we're generating from samples that are sent to the JGI. Next slide. So here's an overview of the workflow, and here the lab, what's happening in the lab is shown in green, and then computational analysis is shown in blue. So generating metagenome drafts is one of our highest throughput products at the JGI. So we're processing thousands of metagenomic samples per year. The workflow that we ended up with combines representation from the lab, informatics, and then the science program leads really represent the users. And the focus here is really on automation. So there's a lot of plate-based work in the lab. And then in terms of on the computational side, we really need defined workflows and processes that don't change, so that we can process thousands of projects a year without having a large labor or overhead. Next slide. This is a detailed figure from a paper that we recently published in M Systems, which shows the details of the workflow. So what you'll see here, if you're familiar with any of these tools, in almost every step we're using a third-party tool that's not developed by us. So really what we were looking at is kind of chaining together these tools, and this so something like WDL is a really good fit. Next slide. So our strategy for portable workflows is very similar to what the NMDC is using. So tasks and workflows are defined using WDL. As Patrick mentioned, sometimes we need to run in different places. So what we've done is we write the WDL as if we're running in Docker, And then if we're running somewhere like Nurse or other places that they might have different container management software. For example, Nurse runs something that's called Shifter. We use a separate config file that can be passed to Cromwell to handle those different environments that swaps out Docker for Shifter or whatever container management tools being used. And then we're using Docker for software management. So a lot of these containers are already available on GitHub and they can just be pulled. We found that this is pretty robust. And then we're using Cromwell to execute these workflows. And that's all I have.
Great, thanks. And we'll move to Kyle, I believe.
Awesome, thanks, Patrick. Hey, everyone. I guess first up, thank you for the invite to come and participate in this panel. Appreciate it. As Patrick mentioned, I'm a researcher at the University of Chicago. I'm also appointed at Argonne as well, and I sort of straddle the two worlds as best I can. In reading the abstract, I thought there were a few systems that we build. So I'm more of a system builder than a workflow developer. We build systems to enable mostly large-scale science problems with large amounts of data or large amounts of compute. So I thought of some of the projects I'm involved in that might be relevant for this this panel. The first being Parsl, which is a parallel programming library for Python that I'll speak about. Globus, which Patrick mentioned before, is a research data management platform. It allows you to very easily move, synchronize, share, even search data in different places. And then FunkX, which is our federated computing platform. So you can think of it as like Globus, but for running functions as service tasks in different places. Next slide, please. So one slide sort of on my background, so you understand probably some of my perspectives throughout the panel. We've built this Parsl programming library for the last few years, probably about 5 now. Our approach has been very much to take a programmatic sort of approach to the problem. So rather than think of workflows as sort of declarative specifications that tend to get obsolete or you tend to need to jam control logic into them, we've thought, well, why not let's start in a programming language where we can enable sort of workflow semantics through constructs in the programming language. The benefits we think are that really workflows are programs and we need to be applying the same types of sort of processes to looking after them, to building them, to versioning them, et cetera, as we do with any other software. Our Parsl library is used relatively broadly. I've listed a few examples of biomedical domains there. Some omics work, but imaging, a lot in places like cosmology and material science and other places. Next slide, please. Okay, and so I was asked to answer a couple of questions here. So again, probably to level set where I'm coming from. How do we define workflows? So in our world, workflows are defined in Python, where there are some constructs that allow you to string together components to create workflows to execute in parallel. We think this is useful because Python's fairly broadly understandable. There's obviously the great language support. You can do almost anything. There's the rich ecosystem of libraries and tools around the edges to help you build up the types of analysis you're doing. And of course, you can leverage common best practices software engineering approaches. What does workflow use look like for us? We work a lot with the national labs, so we run a lot on supercomputers, but we also work with institutional clusters, generally sort of HPC systems. Workflows for us run from a bunch of things that run on single nodes to thousands of nodes, sort of small single core tasks through to multi-node MPI tasks, things that run for short periods of time to longer simulations. Containers, of course, are very popular, but like Alicia just mentioned, we have to deal with these different container technologies on each of the different systems we work with. Who runs these workflows? Primarily, it's people that want to parallelize their codes, often people that start in Python and want to go from their laptop to a supercomputer. Sometimes people that have workflows and want to move to Python because they're struggling with having to throw a bunch of JavaScript into a declarative specification. Our users are very diverse in terms of how often we deal with them. Sometimes they run things once a year, sometimes they run things once a day. And data really is very broad. So I think I've basically said everything in this slide, but we can dig into details as we go. Thanks.
Okay, we move on to Steve.
Thank you, Patrick, and the organizer for the opportunity to be part of this discussion. My name is Steve Sang, and I'm a health scientist and a program officer in the Office of Data Science and Emerging Technologies here at the National Institute of Allergy and Infectious Diseases. So a little bit about our office. The Office of Data Science and Emerging Technologies, or OSDT for short, builds partnerships across NIAID to harness the power of data by coordinating NIAID's data science portfolio and lead the planning and execution of trans-NIAID data science research programs, activities, and related initiatives, both intramurally, meaning within the NIH, and extramurally, meaning with the research community. So about 6 years ago, prior to joining NIAID, I was at the National Cancer Institute, and my work involved the research evaluation of the NCI cloud resources, which I will discuss today. And in that capacity, I developed containerized tools and workflows, mainly for microbiome, metagenomics, pathogen, and microbe-related analyses, and collaborated directly with researchers, users like yourselves, to migrate data analysis to the cloud. So some of the software packages I worked with included, at that time, like software packages like Mother, Chime, Blast, SRA Toolkit, as well as other user-developed tools. So today I'll be sharing with you from that perspective. Next slide, please. As a quick disclaimer, since I was working on the research evaluation in this presentation, those are all my own view and do not necessarily reflect the view of the NIH or the government. Next slide, please. So CRDC, or the NCI, the National Cancer Institute Cancer Research Data Commons, is a cloud-based data science infrastructure that connects petabyte-scale data with analytics tools to allow users to share, integrate, analyze, and visualize NCI-funded research data to drive scientific discovery. The CRDC provides access to data type-specific repositories, as shown in on the right-hand side in those databases in blue, white, and light green, including genomic data, proteomic data, comparative ontology, imaging, and more. So the NCI cloud resources, which shown on the left-hand side of the screen, in the cloud icon there, is a component of the NCI Cancer Research Data Commons that allow users to access those data in the cloud. So cloud resources also provide access to on-demand computational capacities in commercial cloud providers like Google Cloud or Amazon Web Services to allow users to analyze those data. And also cloud resources provide support for data access to a web-based user interface, in addition to a programmatic access using APIs and such. So for me, a lot of what I did at NCI was to develop scientific use cases with end users, leveraging the NCI cloud resources to do microbiome, microbe, and metagenomic related work. And currently, the NCI cloud resources are hosted by the Broad Institute, the Institute for Systems Biology, and the Seven Bridges. Each cloud resource has developed a unique infrastructure with a variety of tools to access, explore, and analyze molecular data. Next slide, please. So I was also asked a few questions here. I'm just answering one. How do we define workflows? Since we have 3 different NCI cloud resources, the definition varied slightly, but the idea is very similar. Let's start from the bottom, the Seven Bridges Cancer Gateway in the cloud. The idea of apps include both tools, which are individual bioinformatics utilities, and workflows, which basically are chains or pipelines of connected tools. And for the Seven Bridges platform, they support the common workflow language. So moving up one, for the Institute for Systems Biology, or ISB Cloud, that platform is intentionally designed to be a lightweight infrastructure on the Google Cloud platform. So it's really workflow specification agnostic, and then you can leverage any workflow manager that's out there. And the users have at their disposal all the tools and technologies that the GCP has to offer. Moving to the top, the Broad Institute, I think Danny will also talk more about this. The Broad Institute of FireCloud or Terra uses pipelines or workflows in workflow description language for the processing of data. So I've included references for all those 3 cloud resources. Feel free to check them out. Thank you very much.
Thanks, Steve. And next we have Danny Park.
Thanks, and thanks so much. My name is Danny Park, and I'm at the Broad Institute. Why don't we go to the next slide? I was asked to say a little bit about kind of what our group does. I'm in the viral genomics group, which is a research group, and I oversee a lot of the computational methods development and analyses in this group. Our group mostly tries to find something at the interface of research and public health where genomics and viral metagenomics informs kind of response to viral outbreaks and things like that. And, you know, usually in years past, even during big outbreaks like Ebola, Zika and things like that, I usually have to kind of explain to folks why viral genomics or metagenomics was actually kind of a useful, applicable thing epidemiologically in these types of scenarios. I actually feel like one thing COVID did is it kind of broadened the general understanding of ways in which genomics can be useful. So I'll move on from here and maybe we'll go to the next slide. A little bit about just from a really high level, what kinds of workflows we have. So over the past decade or so, we've endeavored a lot to move a lot of our kind of in-house research tools for viral genomics and metagenomics into, to get them out of our group, to get them out of the Broad, to get them to our partners. And I'll talk a little bit about that in a second. So, but you know, under the hood, what's happening there, it's the same thing any of the other presenters here have kind of talked about. At least on the viral focus, we have viral assembly tools, particularly de novo tools for a lot of the trickier viruses, but also reference-based assemblies as well. Metagenomics, phylogenetics, things like that. Also tools for, you know, data release, ingest from NMDC, so on and so forth. But as I said, the emphasis here has been kind of on portability and use by some of our collaborators. And currently, yes, we also, you know, obviously we write these in the workflow description language that the Broad uses a lot, or WDL as we like to call it. But the key here is it's been important to us that it actually executes properly on multiple different platforms. So initially for the past, I'd say like 7 or 8 years, our partners were primarily consuming our workflows on the DNA Nexus commercial cloud compute platform. They're a lot like Seven Bridges in a lot of ways. In past couple of years, at least in-house, we've transitioned a lot of these to Terra. We also use CZI's mini WDL for a lot of different, you know, kind of dev and just kind of quick one-off use. But the point is, I think our emphasis has been trying to make sure that these actually do work fine on all those different platforms. Why don't we go to the next slide? So for us, as I said, our primary users for this, a little less than a decade ago, we engaged in an NIH H3 Africa project to help kind of spin up pathogen genomics and metagenomics laboratories in a couple of key sites in West Africa. And so this has kind of launched the next several years of a really fun collaboration of setting up a lot of kind of pathogen genomic capacity in West Africa. And then there's a lot of the work here translated actually to more recent kind of efforts and collaborations we had with public health labs domestically as well. But this is to say, one of the lessons we learned over that time was, you know, although our group probably more so than some of the other groups, we have an emphasis on cloud as the primary platform that we try to execute these workflows on. Part of the reason we did that is that we found that it got it into more people's hands faster and more places faster, especially places that may have not had a lot of the computational or even electrical infrastructure to do it otherwise. But it also just kind of widened the user base even closer to home. So in our own lab, prior to containerizing and cloudifying a lot of these pipelines, you know, a lot of the compute had to be done by the computational people, the experts who are kind of good at that kind of stuff. We found that actually, as we transitioned a lot of this stuff, our wet lab bench scientists are the main people who are asking the questions, the people who are generating the data. They could start actually interrogating it themselves. And so it actually, it widened it. And so the reason I say all this, actually, if you go to the next slide, is, you know, kind of one of the questions is like, how do we choose what to implement, how to implement that kind of stuff? A lot of what exists today from our sets of pipelines are kind of an outcome of several years of just evolution, iteration, and feedback from the people who've been using it in the field, close to home. And a lot of times if there's like a certain project, like a certain outbreak, or even a certain use case, it just highlights, oh, I wish I had this feature. I wish we could change these knobs. We need a new kind of functionality for this. And so that's actually what's driven the kind of software development of a lot of this. So next slide. Oh, the one thing I did want to say, you know, we're not the only ones here in this panel who you know, use GA4GH kind of promoted languages like WDL or CWL or any of these other types of things. But one of the things we did make a point of doing was actually trying to distribute these pipelines through one of GA4GH's tool registry services. So the doc store is kind of the flagship implementation of this. And what this does is, if you're not familiar with it, you can go at some other time, because it's kind of a, you know, but it's a hub not just for, you know, in the same way that like Docker Hub or Quay could be your hub for containers, and GitHub could be your hub for like raw code. What about a place that actually kind of is the hub for the actual containerized workflows themselves, whether they're written in Nextflow or Galaxy or WDL? It can be in this place, and it can actually have integrations. The strength is that it actually has integrations and APIs that allow cloud platforms that know those languages to just automatically pick them up. So you see like this launch with button. You can just click on the DNA Nexus button and pull my pipeline into there, or into Terra, or whatever, that kind of thing. So that's how we, and the other thing that it did is it gave us actually an easier way to give people access to different versions of, because it actually has this sense that like a user can decide they want to pick the latest and greatest, you know, master branch version of the pipeline at all times. They want to run our latest code, or they want to like pin their pipelines to the version 2.1 of what I released a few months ago. Or, you know, if they're asking me about some bug to fix, they can run my dev branch that I just opened up today, or something like that. So that it kind of gave us a conduit to give people access to different versions of stuff like that too. So last slide, I think I was just asked to comment just who uses this. I spoke a little bit about collaborations and how do they run it, what scale. I can't speak actually to like what scale a lot of our collaborators use it as. I do think, actually, I think we have someone from Theogen on the line here who might be able to speak to some of the other use cases domestically, at least. But at least, you know, obviously we use it in our own house. And for us, we have been using these workflows to, you know, the most heavy use has been for our CDC surveillance contract that we've been doing over the past year, where we'll sequence anywhere from 5,000 to 10,000 SARS-CoV-2 genomes per week. So that's like a dozen NovaSeq flow cells that we're analyzing. And that's at a scale where you kind of want to automate as many steps there as you can. And so we did. I can talk a little bit about that later, but that's just to speak to how it's being used. All right, sorry, I don't want to take too much time.
Well, thanks a lot. That was a good wrap up to this session, and a lot of the latter part speaks very clearly to some of my group that have been using essentially the same suite of tools. So I think all the speakers, you guys have done a wonderful job navigating actually a variety of different kind of use cases, and probably represent a large swath of different attendees. So again, I'll welcome everyone online to participate by going to the shared notes document. Marissa, if you can post that again, you can post your questions there. You can also do it in the chat. And I think I'll just start off, you know, with a general question for most of you. You know, just like with anything else, you're using workflows in a sometimes in a very specific manner, like you develop one type of workflow for a particular situation or a particular project. So it's hard to ask anything that really covers broadly everyone outside of, do you see like room for improvement? Danny spoke of, this iterative, I wish I had these extra buttons and bells and whistles to this particular workflow, and you just iterate on it. That's like improving code or improving an algorithm, but is there something broader in terms of distribution of workflows? If you've managed to distribute your workflow, either using something like one of these hubs where you can distribute the workflow itself, or if you just do it in containers, or is there any lessons learned or better ways to disseminate, kind of a standardized way to process data that you've learned over the years throughout your own trials and tribulations? And I think we can just start with the order of presentations, if that's okay with everyone, or you can pass if you don't, if the answer has already been covered by others.
Yeah, I can start. We're just using GitLab right now to share our existing WDLs. I do think there's some work to do in terms of standardization on tasks so that they can be reused between workflows, in terms of, importing tasks. And we've been pretty happy with WDL overall.
So in terms of improvement, I think there's lots of areas for improvement. I think containers have actually been a really sort of needle moving type technology for us, especially with the widespread adoption and the familiarity that researchers have gained with containers. I think they're now fairly comfortable for people to use. I would echo what Alicia just said around versioning GitHub. So as a sort of mechanism, it's again widely adopted. I think we should be looking at methods that are sort of broadly adopted elsewhere and trying to use them for this type of purpose. But in terms of sort of workflow specific problems, I think portability is still a fairly significant challenge, especially in the DOE world, which I understand many people here are from the DOE world. I mean, moving between computing facilities is not nearly as easy as you would imagine it to be, even if you're told it's going to be easy with whatever you're using. Container technology between systems is different. How you even build containers on these systems, depending on underlying hardware, system level stuff, MPI libraries, etc. Portability is a bit of a dream to some extent in that world, but I think we are starting to at least move things a little bit further. I think things around continuous integration and testing in these different systems is a pretty big problem. I think workflow systems themselves probably have very little faith that their system or the configuration for a system will work on any given day in a particular computing facility outside of, say, an environment where you have very tight control, schedulers change, options on schedulers change, configurations, etc. So I think pooling knowledge there would really help us in terms of portability. I think it's probably enough of a rant from me. I'll pass to the next person.
Thanks, Kyle. So from my perspective, I work a lot with end users, as I said in my introduction. And a lot of those end users are less computational savvy than probably most of the audience here today. So, but from my perspective, I noticed there's a very steep learning curve, either getting into containers or getting into a computational workflows. It can be really daunting for someone who's new to the field. So for example, for containers, even for those who have some understanding of containerization, if you really want to scale it up, managing or orchestrating containers for multiple microservices can still be very challenging. So for me, I think the number one thing I've learned throughout the years is that there's a very steep learning curve. The other one I've also noticed is documentation. That's a little bit related to Kyle's point about continuous integration and just putting your tools, code, or even workflow in a shared environment. How well can you how well do you document it? What are some of the best practices in terms of what's included in the documentation? A lot of times what we found is it's really not clear if you're trying to use other people's containers, what exactly is included in there, what version of everything, and how the tools are version controlled. So to me, that's also a big challenge. And also for containerization as a technology, we all are aware that security is an issue. You have to run Docker, for example, as root, and you cannot run that on a lot of actually probably most of the HPCs. So exactly how secure containers are and what kind of data that you're analyzing, especially for control access data, that's also a challenge that we faced.
I'll just say that I ended with a little just bit of description of how we're kind of distributing it and trying to achieve a certain amount of portability. Now, obviously, the extent to which that actually reaches the ideal requires a lot of kind of end user testing and as was mentioned, is not actually that easy to automate, and it's partly just kind of understanding what the rough edges look like when you try to run this on that platform versus that platform and that kind of thing. So, but I think in terms of just kind of the reuse and the getting it into a wider user base and things like that, I think what I've found is that we can work pretty well with the collaborators we work very closely with and have like the ear of, but then when in this past year, as we started to deploy to a larger audience domestically in U.S. public health labs, we partnered with, I mentioned this before, Theogen Genomics, and a lot of the work there was actually translating some of this stuff, like kind of the user's inputs and outputs and just the feel of it, where the guts may actually be all kind of the same. But the user expectations of like, well, I have this file or I want that file. And so it's this kind of funny thing where, and it's, I'm thinking of what Alicia said, where it's like, wish there was a way to somehow commonly just maintain or share the tasks themselves, like the little unit pieces that actually are the same under the hood for a lot of us. And we shouldn't have to independently maintain, but the dressing on it, like the actual way that you thread it together, understandably might be different for this lab or that user because they always like to run this step or that, or they always so yeah, that's just
Well, thanks. Yeah, there are now a few questions coming up. I'll encourage others to chime in or do +1 on any questions you really want to hear. I'll read one of them in the chat. And it's regarding versioning. So versioning workflows, parameters, making them portable is one thing, but how do people approach versioning, if you will, of data, which the workflows work on and the data that's being produced by the workflows?
That's a $1 million question in a way, right? I mean, I assume that's kind of like reference data, databases and things like that, which a lot of these tools will depend on, whether it's a metagenomic data, BLAST database, or some, reference genome, or some other kind of reference set.
Yeah, I'll clarify that question. So this is Robert Michael. I'm Chief Data Architect at ORNL and previously came from St. Jude Children's Research Hospital, where I was leading a bioinformatics group, and we actually were developing workflows right on the cusp of WDL and CWL all coming out and stuff. So good to see the progress here. And definitely one of the issues that we came across of was actually making sure that whatever species, or if it's human, if it's DNA, exome, anything like that, setting up automation to be able to make sure that these things, as they're automated, as jobs are finished, as mapping is finished, QC is finished, any kind of downstream calling is finished, that these things are well categorized, has metadata attached to it, and then as we have top off, or if we have any contamination and have to resequence or something like that, that's all done. And it was a lot of work to make that, quote unquote, automated. My team did a whole lot of that automation behind the scenes. I'm just wondering what the best practices are today, and if there are some more widespread solutions to that.
I can start, I guess. So we have an internal tracking system which assigns IDs and then analysis that like flows, wraps up to a higher ID that's the same. So if we have an assembly and an annotation, those have different IDs for those steps, but then they share an ID as it's, you know, work that's grouped together. In terms of metadata, we're using a Mongo database. So it's something internal that's called JAMA, which I think maybe we're trying to make open source, but we use that to track any and all metadata. And we found that having it not be a, you know, an SQL database makes it very flexible. But then there's also a lot of overhead in terms of you have to be careful about what you name your keys, and that different groups aren't using different names for keys that actually mean the same thing. So it's kind of a double-edged sword there, but we found that that's actually really powerful. And as part of that, we're specifying inputs and outputs in the metadata. And so that helps with data provenance.
I'll just say that I think my read on observing how a few different kind of platforms do it, you know, I think it's seen as an orthogonal problem to the workflows themselves, like that there's a certain data architecture that needs to be built that tracks provenance, that does all those types of things. And it's outside of the workflow code itself. It's part of the system that you're running the workflow code in.
And I guess that's I'll say one more thing and I'll show that, but I guess that was kind of the question. You hit the nail on the head there. Is I was always a big, you know, proponent of the workflows should really be metadata associated with the data, right? So I mean, like you should have, you know, the input parameters you use for cutoff or anything like that to actually, you know, once you create your data, the workflow may change over time, and so you have to, you know, just like you have to track, use CI/CD for your workflows, the data should have the workflows that were used along with it to be referenced forward. So I guess that's where, to me, it is orthogonal, but I guess I'm kind of raising the question, like, should they be, and are people approaching that problem? But maybe that's a bit too off topic, and if that's a different conversation, then that's fine.
I think you're right that it's at the intersection in a way. It's, in my mind, part of the implementation of the execution engine or whatever is orchestrating it is somewhat responsible for at least providing a user the ability to trace those things through the versions of the workflows, you know, the containers, the whatever, that the inputs and things like that. And a lot of them do have the ability, because a lot of these execution engines are oftentimes doing like little call catching tricks. And so they're actually tracking a number of these things. Are they exposing all of it to the user in a way that you can grab at it, is kind of the question.
I personally think your question's entirely within scope. So tracking versioning and provenance is essential. It dovetails in one of my other canned questions, which essentially is, you know, when is it necessary to recompute your data? And that has a lot to do with some of the native, you know, the majority of all bioinformatic tools are comparative in nature. And so when the reference database is changed, then all of a sudden your analyses could potentially change as well. I know that's a really tough question to answer. In your case, you know, I think for provenance, whenever you're validating samples and results, you know, I think we should start at the sample level where you're tracking the samples from which the data comes from. And if you're redoing analysis on the same sample or another process on the same sample, as long as you can link it back there, that would be good practice. Metadata for every workflow and every reference database, so the version control, all that is exceedingly important. Anyway, if any of you want to comment to this larger question of should you recompute data, I would be interested in knowing.
For me, I can probably share some, not exactly to your question, but a little bit about the question that was posed earlier. And I think Danny had a great point about, really, a lot of that is on the design of the platform. On the Seven Bridges platform, for example, data, the platform is basically organized in different projects. And within the project, you can add all the data you need, and then you can run the compute. You're able to track every input, output, and provenance, and also intermediate files with tagging features, various other features that allow you to kind of trace along where those files are produced at which step of the workflow. So it's getting there. It's not perfect, but there are solutions out there, I think.
Anyone else? Yeah, Alicia.
Yeah, I don't think anyone wants to take the bait on your question, Patrick, but I'll just say in general, at least for assembly, if there's just kind of a minor version change, we don't generally go back and redo everything. When new versions of, PPAM or whatever come out, they do try and reprocess those. Generally, we have plenty of work to do going forward, so we try not to make that mountain of work even bigger by going back and redoing everything. So it just kind of depends, but it is a big issue, especially with metagenomics, when you want to do a lot of comparative analysis. So it's really a balance that we're trying to figure out, especially with things like time series. Like, things are changing all the time with the instruments and the library protocols. So I think it's, in some ways, it's not, if you have something like a really big time series, that's just something that you have to take into account, because there's no way that there's variables there. So there's kind of no avoiding it.
Does anyone here have a procedure to do this? It's kind of like a comparative metagenomic effort where you're trying to assess, how things change either over time throughout your samples, or when you're recomputing it, if with either a different database or with a new version of a tool that's said to be, much better in some way.
Are you saying, do we like actually do the regression test ourselves when there's a new?
Yeah.
Yeah, that not so much. I mean, I think a lot of times I kind of hope and trust that whoever's handing down the new mandated version of the database did their work. And similar to Alicia, like we'll rerun old stuff if we have to for the project's sake. A lot of times, for the public good's sake, once our data is out and released in the world, the primary data, then it's kind of on everyone else if they want to reinterpret it, right? But yeah, we don't have a process for that. It sounds like the right thing to do, though.
Okay, we may only have time for one more question, but there is a question about the barrier to entry for the average biologist or researcher, I guess, and for any IT person unfamiliar with either the specifics of what are containers or workflow managers that are being espoused by any one group, plus all the dependencies associated with whatever queuing system or anything that's specific to the Institute. Is there, what solutions exist right now, or is there any solution to make this a bit more seamless for the community, both to jump in as well as to keep current with the best practices, or is it just evolving so fast that it's you learn something and then you'll forget it in a few months because we've moved on since then?
I mean, I'll just say that it's one thing if there's a barrier to entry to people writing pipelines and workflows, but I think that's a little different. It sounds like this is more a question about consuming and using and executing workflows. And I think that's exactly why we, for a while, had initially favored cloud platforms, knowing that these pipelines can run elsewhere too, especially for places that had limited ability to support that. There's numerous commercial cloud vendors that it's just, having your credit card and just start computing, or And part of it is just some of the training sessions kind of walking people through, like, it can be pretty point and click for certain types of routine analyses, and it doesn't actually have to cost that much. But that's just to get started.
Yeah, I think from my perspective, I'll be very quick. It's important to get as close as possible to the people that are actually running the workflow, both in terms of how you integrate with the tools and environments in which they're familiar with working with, and also how you engage with them on a lot of these types of development activities are very much pairwise or collaborative sort of engagements. And I think it's fairly important to really work closely with the people that are going to be running or building these things.
I just wanted to add from the perspective of NCI Cloud Resources. I think all three cloud resources has very extensive documentation on how to get started using containers, using workflows on their platforms. And we understand that moving from like hello world examples to production level analysis is really not that straightforward, but it's very useful in my opinion to have those examples for folks who are new to this to kind of understand what's needed to start running analysis. And the other thing is to include benchmark information, especially for cloud computing. Cost is a huge issue. People who are new to cloud computing, they have no idea how much it's going to cost to run a particular RNA-seq example or workflow. So on some of our cloud resources, they include information such as what kind of VM you should use, or you can use, and that's incur how much cost. So I think that type of information is going to help people get started.
I'm glad you brought that last point up. I do think this is key to the average researcher not involved in kind of a standard, people are now involved in SARS-CoV-2, maybe they have a solution in place, cloud or other. But if you're talking to an average researcher with a brand new project that's generating a ton of data, where do they bring that data? How much will it cost them? And I think that's a consideration that's not often discussed when we're discussing cloud computing. It's sure it's a solution. It's just, it's unclear often, like how long will things run? You're running a metagenome assembly. This is hard to estimate. There are estimators out there, but even they take some compute. So, so Marissa, do you mind putting up that last slide? So, I'd like to thank our panelists again. I believe that this topic is actually extremely broad and could cover, obviously well more than this small hour that we've given it. But I thank all the speakers for sharing some of their insights with us. We will continue this conversation online. The notes document or the shared document is available to continue asking questions if you have. You can feel free to sign up to NMDC. We do care about this community engagement quite a bit. We have a pretty large team that focuses on this. Like Kyle had mentioned, I do think it's important to interact with the actual users to see what is actually the most useful for each and every project or group. The next community conversation will happen February 2nd around science gateways. Please join us signing up to our newsletter or becoming an NMDC champion. And I think that's it for today. Thank you very much.
Thanks everyone.
Thanks all.